Hello and welcome back. Today we're going to be looking more into primers. If you've done the Amino Labs PCR-it Kit, you should be familiar with primers and DNA templates, and you may have also gone through the Amino Labs Bioinformatics tutorial, which shows how to use some open source software called APE to model the fragment size that would be amplified from the PCR-it Kit reaction. And also you did a in silico agarose gel where you could see how theoretically, if you successfully amplified that fragment, where it would run in a gel. So today we're going to continue forward with this topic. In the PCR-it Kit, you were given primer sequences and primers. In this tutorial, we're going to look at how are those primers actually designed and where did they come from. So similar to before, we're going to be using APE, a plasmid editor. Big shout out to Wayne Davis. This is a fantastic piece of software that's helping us to learn about DNA sequences, and it's helping us to design our very first DNA sequences. So thank you very much for doing this. And if you're a viewer here, please consider donating to this project. Okay, so I've got APE open here. Today we're going to be working with a plasmid you should be familiar with already, PUC19. So I'm going to double click on that and drag it onto this screen here. You'll see some forward and reverse primers here. That was from the PCR-it Kit activity. So we're going to design some different primers here. Let me get this a little bigger here. Great. So PUC19 is a DNA plasmid. I'll just quickly click here and you can see it's a circular piece of DNA. It's got a number of different components in it. Again, all these colors that we see in the sequence and we see in the map here, those are annotations that scientists have added to this DNA, and they're regions that have a particular function. Okay, and today we're particularly interested in the yellow regions, or this yellow region here, because that designates as a coding sequence, right? And you'll remember from chapters 4 and 5 of the Zero to Hero book, when you learned about transcription and translation, that a coding region is a segment of DNA that the cell can actually read and interpret and ultimately create a protein sequence. Okay, and so for today we're going to design primers to amplify a region of this plasmid that codes for a protein called LAC-Z alpha. Okay, so I'm going to go through this top area here. I'm going to just click on the LAC-Z alpha and you'll see it highlighted. The sequence here will automatically become highlighted. We can see that the sequence starts with an ATG, which you should know codes for methionine, which is always the starter amino acid of protein sequences. And it ends in a TAG, which codes for a, that's right, stop codon, which designates the end of a protein sequence. Okay, so this is highlighted. We can go ahead and double check that it is in fact a nice cohesive protein sequence with no stop codons. I'm going to click on the translate feature here. I just need to click down here so that it comes up. And I can translate the selected sequence, which is for the LAC-Z, or you'll see that it actually populates for any of these yellow regions which designate them as coding sequences. I'm just going to leave it on the selection. I want to see the one-letter amino acid code, and I want to have the DNA sequence above that. I'm just going to click OK. That's going to open on another monitor. I'll drag it up here, and we can see the ATG again starts with the methionine. The TAG is a nice little star for a stop codon. And we can see that there are no little stars throughout this sequence, which means this will be a nice protein. It'll start with the methionine and it'll go all the way through and end with the stop codon. All right. Sometimes if you're trying to amplify a coding sequence, It could happen that there will be a star in between somewhere, which is not good, and that means you need to look a little more into your sequence. Okay, so now let's get into how do we actually design primers. And there are lots of different software that'll help you to do this. You could search in a search engine primer designer and find something. I think Ape is really good. It's got a very simple way to do it that works most of the time. And I'm going to show you how to do that. So I'm just going to click to unselect this region here, but I'm keeping an eye on where it starts and ends. I'm going to start highlighting from the beginning ATG here. And as I do that, you're going to see up here two values changing. Well, a lot of values changing, but these in particular are important. The TM stands for melting temperature. Okay. And the melting temperature is the temperature at which your primer is no longer going to bind or anneal to your DNA template. Okay, and basically at this temperature, there's enough thermal energy to break the hydrogen bonds between the nucleotides in the primer and in the template, and so that primer will then fall off. Okay. And so when we're doing a PCR reaction, you might remember that in phase 2 of the reaction, there is a step called the annealing step. And that annealing step temperature is correlated to this melting temperature of your actual primers. Okay. We want to strive to get about 60 degrees Celsius. That's a pretty good melting temperature in general for primers. Although as you become more pro at doing PCR, you'll encounter situations where you want to have higher TMs or you want to have lower TMs. So in general, 60 is good, but you may ultimately in the future want to design your primers to have different melting temperatures. Then on the right here, we have %GC, which is the amount of G's and C's within the sequence you're selecting. In general, we want to try to get 50% so that our sequences have 50% GC and 50% A's and T's. A range of 45 to 55, if you can be within that is great. 40 to 60 is okay. It still should work. But beyond that, it becomes difficult for the companies that you send the sequences to actually synthesize your DNA. Okay, so ideally you want to design for in between 45 and 55 and have 40 and 60 kind of as the outer limit of what's possible. Okay, so I'm just going to go through and let's watch those values as I select more and more. And again, we're trying to get to a TM of about 60 and a GC of around 50. So one by one here, 58.8, 59.6. That's very close for a TM. That's very good. And the GC is within that 45 to 55. So this is a very good sequence. That should work really well. If I go one more here up, we've got 61.9 and 48. So I would say while the GC is closer, the TM is a little bit high. We're at 62 now almost. So let's dial that back to this 59.6. So this is a very good sequence. So I'm going to copy that just by doing Command or Control C. I'm going to open a new sequence here by doing Command or Control N, or you can go up into the file at the top and just get a new sequence. I'm going to drag that up from another screen here, and I'm going to paste. So Command Control V. All right. So that is great. We've got our first sequence, and I'm going to let's do something important first so that the software knows that this is a primer. Let's highlight that sequence and right click, and we need to create a new feature. So that opened up a window on another monitor. Here it is. And that new feature is that this is a primer, right? So I'm going to click down here. Right now it's listed as miscellaneous feature. but we want to go to, so we've got lots of different possible purposes of this sequence, and it's not a gene. Whoops, it's and then if we go here, it's not a signal, but when it comes to binding, this is a primer binding region. Okay, so this is a way to designate this DNA sequence as a primer, and we're going to call this the PUC19 underscore lac Z alpha. I'm going to use a little a here for the alpha because I don't have the alpha character. And we're going to put FWD, which stands for forward, because this is at the beginning. It's at the 5' end of the coding sequence. And I'm going to click OK. So now we've got the annotation as the lac Z forward primer. But then I also want to save this DNA sequence. So I'm just going to now click Command or Control S, and that's going to pop up the normal save box. And I'm going to call it the same thing. I'm going to call it the PUC19 underscore lac Z alpha underscore FWD for forward. There we go. So that's saved. I'm going to move that over to the other side. And now let's design the reverse primer. So very similar activity. We're going to find the end of the lac Z coding sequence, which is this TAG here, and we're going to highlight our way backwards and see if we can get a nice primer. TM 55, 58, 58, oops, 58, 59.5, 61, 59.5. So 59.5 is close to our other primer, and it's close to 60 degrees. And look, the GC content is really nice. It's very close to 50. So this here is a very good primer as well that matches our forward primer from the standpoint of the melting temperature. So this is great. So I'm going to do the same thing. I'm going to copy it. And I noticed I've got another window open here. So I'm just going to paste it here. If you don't have another window open, just do the same thing as before and open it. Now, we do have to do one more extra step for this reverse primer. Okay, so you'll remember, again, going back to chapter 4 of the Zero to Hero book, that DNA has direction, right? It's asymmetric. So the plus strand will go from its 5' to 3' direction, and the minus strand has the opposite 5' to 3' direction, right? And so this primer is designed to match the forward or plus strand. And so we actually need to change this reverse primer so that it's the complementary, it's the reverse complement, okay? And in order to do that, you can use a very simple feature where you go to the edit in the toolbar above, and you go down and you'll see reverse complement. Okay, so I'm going to click on that, and you'll see this sequence change. There we go. So now this sequence is changed so that it's no longer this highlighted sequence, but it's the reverse complement of this sequence, which basically means you'll see here we've got CTAT, which will bind to this GATA. Okay, so you'll see that this is actually binding to the opposite strand and going this way, pointing backwards towards the 5' end here, so the forward primer. Okay, great. So we're going to do the same thing. We're going to highlight that. We're going to create a new feature. That popped up in another monitor. That feature is binding. It's primer. And we're going to change the name here to PUC19 underscore lac Z alpha underscore reverse. And click OK. Fantastic. And then I'm going to save this as before. Same name, puck19 underscore lac z alpha underscore reverse. There we go. So now we've gone through, we've loaded and looked at our template, our puck19 template. We've used APE to highlight some sequences to try and find primer sequences that have a nice TM of around 60 and a GC content that's close to 50%. We did that. Sometimes it takes a little bit of wiggling. Sometimes there's compromise that has to come into play. And unfortunately, you have to do that sometimes. But as long as your forward and reverse primers have similar properties, usually within 3 degrees melting temperature, then your PCR reaction will generally work. Okay? Good. So now we've got these. Let's use the PCR feature that you might have experience with already to model and confirm that these primers were designed properly and that they'll actually create a PCR product. So let's go up here to the tools. Let's go down to PCR reaction. It's going to open up that familiar window where we can see the linear visualization of our DNA template. We can see here it's properly loaded. We do see the primers, but we want to have our template as the puck19 plasmid. And then we go up to the database primer sequence. Let's click on that one. And it comes up as a forward primer, although it is showing something else kind of funny here. We want to make sure that we're ultimately going to click on our primer here. This probably has to do with some of the other annotations within that sequence. So you can always right click and delete all of the other annotations, and it might simplify this screen a little bit. But yeah, so we've got the reverse primer here. So let's also load the forward primer. There we go. So we've got the forward and reverse primers here, and then this other marker, which let's not let that confuse us, okay? Great. And then we want to select the primer pairs. Let's select the forward here, and let's select the reverse here. Again, let's not select that one. Our template's good. We've got our newly designed primers. Let's click New PCR Product. That opened in a new window, and it successfully opened. Okay, let me just move this over here. All right, I'm just going to close this so I can move this to the front. And we can see that it did successfully provide a sequence output, which means that the primers are designed properly and the PCR reaction would work. If you had chosen the 2 pairs, and let's say you forgot to do the reverse complement of the reverse primer, it would not spit out any sort of sequence, and you would know that something had gone wrong. So this is the beauty of APE and doing these in silico experiments, is they help to do a kind of a checkpoint to make sure you're doing things right. Good. So we've reached our goals today. We can see our LAC-Z sequence. We know that our primers are at a TM of about 60, which means that if you were to do a PCR reaction on this, you would set your annealing temperature a couple of degrees Celsius lower than this, so maybe about 58 or 57, maybe even 56, and that at those annealing temperatures, your primer should bind nicely to your template. Good. So the last step we want to talk about today is simply, where do you get your primers from? So we've gone and designed them. How do we actually get those? And so there are companies out there that synthesize DNA using chemistry. All right. And one such example, which is a long-standing company that provides great product, is IDT. So IDT, which is Integrated DNA Technologies. I'm just going to put DNA here to make sure we get to the right place. And we can see here, IDT. Let's not click on the sponsored link here. Let's just click on their website. There we go. So this is a company that you can send them your sequence and they will synthesize it for you. Now, there are some drawbacks. So biotechnology, as you've probably been hearing, is a very powerful technology. And so often to get DNA synthesized, you need to be a company, or maybe you are in a school, right? So you won't be able to order these if you're at home. There's a very low or no chance that you'll be able to order primers directly to your home. It's a safety measure that's put in place so that nefarious activities don't happen. But yeah, it's as simple as that. You would have an account, you would paste the sequences, copy paste or upload the sequences as text files into their ordering portal. You would tell them the sort of purity you want on your primers, and you would click order and you would pay, and they would come in a few days. The turnaround time for primers is generally very fast, especially with IDT. Great. And then you get those primers. You need to add some ultra water to those, right? So they come as dried samples. You add some ultra water to those to get the right concentration. And then you can add them in your PCR reaction along with your DNA template, and you're good to go. And that's, again, the activity you did in the Amino Labs PCR-it Kit. Great. Well, I hope this tutorial was helpful. There will be more primer design tutorials because there's a lot that you can do in designing primers. But we'll stop there for today. Happy PCR. Happy experimenting.